Good to be seeing all of you. I'm Ursheet. I'm one of the partners at Mayfield. We lead their investments in human and plant health. Uh, Mayfield is one of the founding venture capital firms in the valley. It's been around since the 60s. We partner with entrepreneurs such as Lucas very early, typically seed, Series A, sometimes Series B. It's a lot of joy and pleasure for me to have Lucas in conversation today. Lucas is one of the founders of Mammoth Biosciences. It's been a delight and joy to see them just get it out of the park in the velocity of how they have developed more and more things in the building CRISPR systems and how they are enabling an ecosystem. The title of the topic was updated, essentially talk about sort of CRISPR plus the next generation of CRISPR systems and applications. And that's what we'll kind of touch upon. There will probably be some time for Q&A, so if you have some questions in the final 10, 15 minutes, so please do raise your hands. And so with that, let me start out with something. So there is so Lucas is known by has a nickname, and it's the CRISPR Whisperer. And Lucas, how did that come to be?
Yeah, it's been I think a very rapid trajectory that we've taken since starting Jennifer's lab. But it really started, I think, maybe back with this concept actually that I think we encountered way back in like middle school, which is something you might be familiar with, Pasteur's quadrant, where basically this idea of applied basic research. And I think CRISPR, once I encountered that, once I started at Berkeley in Jennifer's lab, that really was the perfect distillation of this concept, where you're doing very basic research, trying to understand how CRISPR systems, in this case, protect bacteria, and from there, applications just kind of flowed naturally. And I think it's a really unique section of biology where you can do that basic research and have it lead application. So it really snowballed as we started to understand this natural diversity. We were also collaborating with Jill Banfield's lab, which is much more focused on metagenomics and really just mining diversity naturally. And then, from there, we were able to find some really cool new CRISPR systems that unlocked this whole new section of using CRISPR, specifically being able to use them beyond the liver and beyond ex vivo applications. And that was really one of the foundations of Mammoth. And from there, it's just been, basically taking the things that we were doing in Jennifer's lab, which were very, hands-on, and figuring out how do we actually build automation to do that on a scale that's 3 or 4 orders of magnitude beyond what we could do as graduate students in the lab. And here we are today now thinking about how we actually apply those for patients and getting them into the clinic.
So I think Lucas is being very modest because I think the whisperer terms kind of come with, I think, Lucas and his team at Mammoth have probably been one of the most prolific discoverers and developers of new CRISPR systems. So I'd love, Lucas, for you to kind of walk us through just that history, right? Which is, where were things in 2014 when the first set of CRISPR companies were formed? And how has the field kind of developed? And what have been some of the cool new systems that you and your team have developed?
Yeah, definitely. And I'll preface this by saying, from my perspective, it's still very early days for gene editing and for CRISPR. I think we're just scratching the surface of what the promise is. But if we rewind the clock back to 2012 or maybe even before, CRISPR specifically was this kind of scientific backwater that no one was that interested in the mainstream media. And Jennifer definitely was very focused on the RNA biology and how this worked from a basic science perspective. And it really was just happenstance that these tools they were working with were very applicable towards genome editing and that they worked in mammalian cells. So as with a lot of times in science, the first thing that got discovered just kind of got wind behind it, and that started to move forward, and that's really the premise of a lot of these first generation CRISPR companies that are based on Cas9. But that said, I think with the haste to get those things advancing into the clinic, there was definitely a missing piece of what else was out there for gene editing. And CRISPR, there's actually hundreds of thousands of different CRISPR systems that just exist in nature. Probably just the bacteria in this room, there's probably thousands of CRISPR systems. And so, what we really decided to focus on was looking at that diversity and saying, all right, statistically, the chance that we pick the first system and it's the best system, it just seems very improbable. So let's actually understand that diversity, see if we can overcome some of those limitations of the legacy system Cas9. So we originally focused on ultra compact CRISPR systems. We'll probably talk about today, but the biggest challenge with gene editing is how do you actually get it into the right tissues. And these ultra compact systems are much easier to get into the target tissues using delivery like non-viral like LNPs or AAVs. And yeah, from there, we started to build on top of that what we call CRISPR Plus, so actually being able to not just use CRISPR as a pair of scissors, but actually to think about them as more precision tools that you can use to do epigenetic regulation, modification, or more precise gene correction. And I think now we're really looking towards how do we put all these pieces together in terms of delivery technology, in terms of the gene editing systems, to move beyond just editing liver, which is really where, when you see all the progress in the news for gene editing, that's really what we're seeing. Is I would think of it more as a good kind of demonstration of the utility of CRISPR, but falls short of curing most genetic diseases, which is ultimately the promise. So that's really the timeline for the company and for the field overall. And I think going forward, I think it's about progressively being able to treat more and more diseases that moving away from rare genetic diseases and actually thinking about more common and prevalent diseases that have huge unmet needs for patients.
So let's take the ultra compact systems, right? So like, what is Cas9 size, and what would be like maybe top 2 or 3 examples from Mammoth of the ultra compact systems, and what would become sort of the applications that become that unlock these new systems?
Yeah, so the first Cas9 systems, to put some numbers on it, was about 1,300 to 1,400 amino acids. So in terms of bacterial proteins, a very large bacterial protein, not compatible with most of the viral delivery methods that we use. The newer systems that we use, so a few of them would be Cas-V, which was, I think for us it's one of our larger systems, but even that is about half the size of those normal Cas9 systems, so that's about 700 amino acids. And then our most compact systems go down to about 400 amino acids. So you're talking about something that's about a third or less of the size of the original CRISPR systems. And so what that enables, again, going back to the key challenge of how do we actually deliver on this promise for gene editing, is it allows you to use delivery vectors that we have, namely things like AAV, which have a very, very constrained packaging limit, and it allows you to not just fit the CRISPR system, but fit all the other things that are needed for creating the bespoke therapy for a particular target. I think one thing to think about and to keep in mind with CRISPR is that CRISPR is the heart of the therapy, but there's really a whole ecosystem of other things around that that's required to get this to patients, from the genetic understanding to the regulatory elements that you might put on there, and then of course the delivery vector. So when we think about enabling CRISPR, for us it's thinking about how do we actually innovate on that kind of core component, the CRISPR system, so that we can better use all these other components that are required to push a therapy forward.
Yeah, I think that's been one of the sort of things I had, right, because of working with Mammoth and the mindset. So pre-Mammoth, most of the CRISPR companies, right, they started out with Parikh and They went with like Cas9 and they decided to sort of just build the application out of that. And in many ways, you know, it didn't pause to kind of think whether there would be better systems out there, whether they would be the ideal ones. But there was also this tendency you want to kind of keep it just to themselves and do it all on their own. And one of the core founding premises of Mammoth was that, you know, this is going to be a platform company, much more something that enables an ecosystem that we can think of like a Microsoft or PCs, or like an Apple with mobile phones, where there's a core set of things you do well. There's a subset of applications you develop, but you really focus on the few things you do well and then partner with everybody else along the way out on that end. And you know, you develop a wide range of applications, not just the ones that you may think are the most profitable ones out on that end, because there's a broader ecosystem, and that's where diagnostics. So how did you know? And so as you were sort of doing the ultra compact systems on edit, what else did you end up finding, kind of like diagnostics, as potentially interesting applications of CRISPR, and how do they go beyond healthcare? And then I'd love to kind of come back to the epigenetic and the CRISPR plus stuff, but you know, while the ultra compact systems were happening, you were essentially developing CRISPR for beyond therapeutics.
Yeah, for sure. You know, I think at its core, right, CRISPR is a way to program a protein to a sequence of interest, and that can be in the context of a genome, it can be in the context of in vitro diagnostics. So this also goes back to our work back at Berkeley in Jennifer's lab, but one of our co-founders, Janice, she was very interested again in the basic science of how does a CRISPR system cut its DNA target, and in doing that and running, this is, you know, in running radio-labeled gels and very old school biochemistry techniques, we were able to start to see that that some CRISPR systems had this feature where they don't just cut the target that you program them to, but they turn on this collateral activity that allows you to detect a signal. And so that led immediately to you know, development of CRISPR for diagnostics, which was a field that really, you know, hadn't been contemplated before. You know, thinking about this, and that we developed in response to the COVID pandemic as well, for much more rapid detection. But beyond that, you know, I think, when you think about it in the genomic context, you know, we're largely focused on human health internally. With Ursheet's point, you know, the applications of CRISPR go well beyond that. So, of course, you know, many of the companies that you might hear talk are using CRISPR for one application or another. You know, strain engineering is definitely a key area, especially when you're talking about more exotic you know, organisms that don't have the robust molecular biology tools that we have for things like E. coli. And as well as agriculture. I think agriculture is actually where we're going to see probably some of the biggest impacts, most immediate impacts for gene editing. And I think, yeah, it is important for us as a company. This goes back to just the kind of ethos of the company as we were founded, that we actually enable other groups to do this and really kind of build an ecosystem of applications beyond what we can do internally. Because even with our, you know, rapid growth, there's only so much we can do. Even within genetic disease, there's only so much we can do. And so making sure that the tools that we develop get disseminated more broadly, I think is definitely a key, you know, thesis for us as a company.
Yeah, no, I think one of the things that in the early days of Mammoth has kind of come together, the founders came together, the core things was to align on the values and the mission. And the idea being that if the world had X number of CRISPR products 10, 15 years out, with Mammoth it should be 10X or more. And that could only happen if we took an ecosystem and a platform kind of mindset, which was actually pretty different from what generally most people were thinking about in therapeutic life. So going back to, you know, building and developing new CRISPR systems, right? You spoke about size. What are some of the other attributes that have made these CRISPR systems better?
Yeah. So, you know, a lot of, as we think about CRISPR, I think the kind of 1.0 technologies were just making a cut in the genome. In order to be able to do other types of edits, you really need a repertoire of different systems. Size, I think, is one of the foundational things that a lot of things are built upon, but specificity, especially as you move to some of these other applications, is also a very, very important one. When we talk about treating diseases that have have some kind of standard of care already. I think those safety burdens become higher and higher, right, where there's really no tolerance for any off-target cutting that might happen. So mining through that natural diversity, we're able to find systems that were much more accurate. I think just like the cutting mechanism is another thing that we were able to build up. And yeah, I think another key thing that we think about is this portfolio approach. I think, yeah, when you think about Cas9, there's a lot of applications that have been built on that, but there's a lot of applications that just you can't do with that system. And so generally, we don't want every company to have one little area that they can work on, but for us, that we actually have the breadth of tools to be able to widely use these systems, whether it is for epigenetic editing or for editing plants or for diagnostics, but you actually have all of those systems under one roof, and you can actually choose the best system for the job as opposed to just being stuck with whatever you got to start with.
Yeah, I think that is a very interesting and an important attribute, because I think that kind of fundamentally works to the platform model. If you are developing an application, in this case an application can be a diagnostic, therapeutic, or it could be a new food new material plant, you want to be able to use the best tools available to you to kind of solve the problem, rather than only the tool that you had licensed out. And structurally, that is core to why partners find Mammoth to be a really good partner, because they know that the innovation engine at Mammoth will keep on finding more tools, and if they're trying to solve the problem for a given disease, they will be getting the best tools. In contrast, what has happened is that there have been companies that have taken each version of the incremental development of CRISPR and have become sort of dedicated kind of companies. And from a application developer or a therapeutic company perspective, that becomes kind of a big challenge in that they may start working with a partner only to find that the version of the tool they need is actually sitting with somebody else. And then I think from an investor perspective, this is be it private investors or public investors, you sort of end up having to worry about are you picking the right tool, or are you sort of are the people who created your tool all in on this company, or are they onto that next version or the next shiny object? And this is kind of fundamentally this thing about how do you think build to last things for the long haul versus not. So that has been there. So now introduce CRISPR Plus. How does where do you sort of draw the line from all of that, and how that has been working to where the future is headed?
Yeah. So CRISPR Plus is a term we came up with a few years ago to really capture this evolution of CRISPR beyond making double-stranded cuts as a genome editing tool, recognizing that most diseases cannot be cured just making a break in a genome and doing a knockout. And so a lot of this is thinking about CRISPR as this method to search out a sequence, really this kind of homing system, and to fuse on different modules that are going to change the effect that you're getting. So this can be things like epigenetic editing, where you're actually permanently changing the methylation of the genome, which can silence genes, or turning them on. It's also more precise methods to not just correct things that you can knock out, but actually write in sequences that you want. And so, this is really one of the key pieces of how we develop this to more broadly address genetic disease. I think one of the maybe dirty secrets of the CRISPR Plus field, though, is that for a lot of these companies, you're taking a large system, this Cas9 system, and you're now fusing on even more machinery, even more baggage onto those systems. And so you end up with these systems that are so enormous that they really have pretty limited applicability, except in the ex vivo applications where you can just electroporate basically the CRISPR systems into the cells. And so I think it's really been a natural transition for us as a company where we've got these very, very small CRISPR systems which leave open all this payload in traditional delivery vehicles, where we can now actually fit that machinery in and efficiently deliver it to tissues in vivo. Yeah, and I think that's been a, it wasn't the original, just only reason that we developed those small systems, but I think being on the forefront of that, it's really positioned us to be able to push those forward into the clinic and not just being research tools.
Yeah. So as you kind of look at the crystal ball sort of going forward, right, what are sort of the diseases that you think we will have therapies in the clinic in the next 3 to 5 years?
Yeah, so of course, the field overall, I think most are closely watching and anticipating that we'll have the first commercial sickle cell programs. Again, I think a very exciting milestone for the field, but still in an ex vivo application. I think we're seeing a lot of activity around rare genetic disease in the liver, but I think really where we as a field want to be moving is towards these much more prevalent, serious, unaddressed diseases, especially as we think about degenerative diseases in the brain, the CNS, or skeletal muscle, or the heart. And I think it's also eventually as we get these proof points, especially with rare disease, I think it's about moving more towards preventative medicine, right? So things that maybe aren't as acute, but that we know 10 years down the line or 20 years down the line, you're going to have some kind of adverse health consequence. Necessarily we need to build up the safety profiles of the systems before moving into those, but that's really what gets me excited. And for me, a lot of the initial programs, for both Mammoth and otherwise, are really to try and develop the systems, get those proof points on the board, so you can go after things like curing Alzheimer's or Parkinson's, things that many people have had personal experiences seeing the impact of those.
So you're saying that starting out with the ex vivo stuff includes some blood-based diseases like sickle cell, but also of course this is the CAR T engineering, and then that's now moving next step to the liver, and I think LNP delivery with mRNA is probably providing a good sort of accelerator out on that end, and then from there it is to sort of this is like DMD on the muscular side, sort of more CNS, and then the big thing being even sort of taking on kind of chronic issues like cholesterol and sort of obesity and that sort of thing. So it is definitely sort of very, very fabulous. So Lucas, what's the secret? How is it that Mammoth's able to do these things so much better? And like clearly the conversation that everyone's kind of talking about is generative AI, large language models, and how AI is going to turbocharge or change everything. And I'm going to hear a little bit about what enables your success in this level of sort of research and research productivity.
Yeah.
And how much of it is technology, how much of it is culture, and what kind of technology?
Yeah, I think a big part of it is the team that we've been able to assemble, as well as the commitment to innovation. I think going back to their early commentary, I think there is a tendency for some companies to take something that's working and just kind of harvest it, just think about how to monetize it. I think we definitely take a longer term perspective, and this isn't just the internal management team, but I think it does require a cohort of investors that are supportive of that and that are thinking on long time scales of how do we make an impact on the species. So I think that's kind of the foundation, and then to think about it more technically, a lot of it's just the science that goes on internally. Going back to how we were doing this even in an academic setting, a lot of it is very, very creative approaches to finding these systems. So the models that you use, I think there's of course a lot of excitement around AI and LLMs recently, but I think most of us know in biology that these genomic data sets are really built for machine learning, and it's just kind of something that just happens naturally that we use these tools and have developed these tools. And so, I think that's been a key part of it. As with all, any AI approach or ML approach, it's about having very clean, good data sets to start with, and that really was the foundation. Is this metagenomic engine that started this, and now it's much more around protein engineering and continuing to evolve and adapt the systems. But I think technical things aside, a lot of it is the culture of the company, of really what we set out to do and keeping that vision in mind as we continue to grow and not letting our success kind of distract from the long-term vision.
I think you brought up the culture and the long-term sort of mindset view, right? Like, changing gears, what has been your growth journey from scientist to sort of entrepreneur, manager, leader? And so I'd love to kind of get some look back on your journey and both what are your learnings and what is your advice for anyone that's kind of really take the benchtop science and translate that into real products.
Yeah. It's definitely been an interesting and awesome opportunity to be able to do this. I think thinking most of the time that people spend in graduate school doesn't prepare you to actually manage and interact with people. I think that's you learn the science, you're a technical expert, but a lot of it is progressively, and by no means are me or any of the founders perfect at this, but progressively getting better and better at orchestrating the team and really taking that scientific knowledge, using that to actually motivate and build credibility with the group. I think it is a challenge, and not all PhDs make it through that hurdle. And I think one of the big areas that probably we should adapt the education system, the graduate education system, is a much bigger focus on just communication generally. I think it's important for entrepreneurship and of course leading teams and building teams, but it's really important for of course anything we do pretty much, and just also educating the broader public about what the opportunity is of the tools that we're building or the broader SynBio community. So for us, it's just been also of course working with mentors and having coaching, being open to feedback, and really taking that feedback to heart and iterating.
And so as you sort of, and I see you kind of built a good team, you partnered with great people. How do you sort of, what do you look for when you are trying to, what is your process when you kind of are trying to recruit or bring in someone, and how do you start with like benchmarking excellence for the role? Because there's so much that you don't, right? Like in our opinion, the company is fundamentally limited by the learning ability and agility of the founders. And so the founders really end up having to kind of, independent of the role they take within the company, their influence is really broad, and it does require them to kind of really develop broad aperture and effectively build their own infrastructure for learning very quickly. And so I'd love to hear on what is your sort of personal process. And then what do you look for?
I think early on in the company, when didn't have much experience in hiring, I think my initial instinct was to just hire the people that seemed the smartest, just the technical experts. I think it is, if you just push a bunch of technical experts together without actually curating that group, I think it very quickly crumbles and falls apart. So I think a lot of it is hiring, maybe indexing less now on that and indexing more on, is the person actually passionate about the company? Are they going to be committed? Are they agile and able to learn quickly? Because most of the stuff we're doing, you're not going to find someone off the shelf that's done it before. And then of course, are they going to work well within a group and actually be able to build something bigger than them as an individual. Often, I'll in interviews ask someone, especially given that founders, I think our job is not to be the technical expert. Of course, we've got some of that background, but it's really to bring in those people that can educate and actually help to be this additive effect. So often, ask someone something that I have a good sense of, but just kind of teach me about this and how they respond to that and how eager they are to really dig into what they know. I think is a good signal for how well they're going to be able to adapt to a startup like Mammoth.
So we have about 10 minutes left. I can continue asking lots of questions, but if any of you guys have questions, raise your hands. Go ahead.
Thank you very much, Lucas. So now I'm quoting Jennifer. She said that a cure is not a cure if people that need it, they cannot afford it.
Yeah.
So which are the bottlenecks to get to the point that everyone can afford CRISPR based therapies?
It's definitely it's not an easily solvable challenge for sure. I think it is important to have that perspective over the lifetime of the technology as well. I think the kind of build the roadster first model and then try and build Model 3 afterwards is definitely something that I think resonates here. But I think it's also about it's not just a question about the intervention. I think a lot of this is that the healthcare system is built to have this chronic care model where you're giving someone a pill every morning or giving them an injection every month. And I think CRISPR does have the potential in terms of the curative things to actually condense that treatment cycle into one treatment. I think the biggest cost factor is going to be also about the regulatory strategy of how do you get multiple successive things into the clinic without having to reinvent the wheel. So actually being able to build this as a platform. Of course, the first program, again, the Roadster will be expensive, but those successive ones should be able to stand on that. And that's not, the FDA and CBER, they're very smart people and they're trying to figure this out and grapple with it, but I think there needs to be some kind of platform approval process that also allows you to move into rarer and rarer genetic diseases. So no silver bullet, but I think, of course, as everyone knows in healthcare, the cost is usually not the cogs of the therapy, but it's actually all the investment that goes in up front. And a lot of that is driven by what's required by the regulatory process to prove that it's safe and efficacious. And so thinking more creatively about that, especially after we have these initial successes, I think is going to be important to driving the cost down of these therapies.
Can you expand on that at all on the regulatory side, or is the FDA engaging genome editing platforms and asking how they can make the platform power of it for their applications?
I think they're willing to talk about it now. I think it's still too early in the technology life cycle for them to be thinking about that because it's still this one-off thing. But the ideal situation, even if you look at one genetic disease, you often have mutations scattered across the disease target. It's really rare that you have just like one perfect mutation, and that's of course what all most of the programs are focused on. And so I think what you want is a way to get kind of a broader approval for the platform, and then CRISPR, it being a platform, you can just change out the guide, and having some kind of minimal safety requirement of actually understanding that guide is actually the same as in terms of safety from the perspective of off targets and things as the other guide. And then being able to get a kind of leg up on the program and actually more broadly address the patient population. So I think it's still too early, and whether it's the FDA or MedSafe or EMA, I think that drives this. It's going to play out, of course, as most regulatory policies do over the course of years and years. But I think all of that said, I think the access issue is definitely important, but there's also treatments that need to be delivered immediately, and that's the first step that needs to be taken, even to broaden that access subsequently.
Yeah, I mean, I think I remember one conversation with Mammoth team, as COVID was beginning to happen, it's like, hey, there was that kind of team really focuses plans as, wants accountability, and they had a plan for the quarter, right? And come January, a side project started, a paper got kind of published, and then it was like, well, we don't really know what the regulatory plan will be. It's like, don't worry about it, just go do it. And even, and we saw, right, like the speed with which things in diagnostics sort of, got the emergency use approved and things like that. When I'm looking at regulators, this is what I advise everyone to think about. You got to kind of go ahead and recognize that they have a hard job. They don't get rules to bring to market fast, but they sort of do end up having to take a lot of the pain for anything that's done prematurely. Having said that, they went to do this not because they want to stop you. They essentially, most of the regulators, they're scientists, they're engineers, they kind of care. They want things to get to people. That's why they enter that field, right? And so we do make a common mistake that a lot of people do is not talk to regulators early enough. And because if you really think about it, one of the best things you can do is develop a direct connect, right? If this is negative news, you might as well get it good. You want to sort of go ahead and engage in that kind of conversation. And over time, you do see sort of good change in mindset emerging here. And often what happens is I think emerging companies actually make that as a bigger mistake than some of the larger kind of companies, because the larger companies will often have a lot of dedicated people who are talking to regulators, so they get to influence things. The advantage you have as an emerging company is that your senior leadership and your best scientists can actually engage in the conversation. And so the regulators actually like talking to you because they are not talking to layers and layers of corporate BS, right? They are actually talking to people who will acknowledge, help them understand, right, breaking the core issues out around that. So I think I wish there was more regulatories, for example, on the tech side of the house, like given how dangerous AI has already been, like you see what TikTok's doing to teen aggression, and in teen suicide. I mean, you've seen like the number of young adults, like death rate has gone up dramatically in the last 5 years. I wish the rest of the world actually took a more sort of regulated approach to this, the way healthcare does. You had a question?
Yeah, so, excuse me. So, Lucas, you talked about how AAV imposes delivery constraints on how big of Cas9 you can deliver. Could you alleviate that by using mRNA? I mean, mRNA and LNP, now they're talking about delivering, self-replicating RNA about 1,000 nucleotides long.
Yeah, I think it's a great technology. I think the challenge there, especially when we're talking about programs in the clinic, is it's really constrained to the liver, and it's a pretty small subset of diseases that the liver is a great place to target. It's kind of this production place for a lot of things. But that said, it's still pretty constrained, and viral delivery, specifically AAVs, are still really the gold standard for everything extrahepatic. I think this is delivery. If I'm completely honest, delivery from the technical hurdles is more challenging than gene editing. Right? We build something that goes and cuts a sequence in the genome, and it's great for us that we've got the easy task. But delivery is really hard to get systems that work, effectively and safely across different models as well. And so our perspective has been, we'll be agnostic in terms of the delivery technology, and as let's say LNPs mature and hopefully you can move extrahepatically, we're ready to use those. But what we have today is the viral delivery vectors, and in terms of the treatments that we want to push forward, let's take what we've got versus compounding risk with a bunch of new elements that are going to be more risky altogether.
Well, we have time for one last question, and if no one has one, then I'll take that question. Well, you have the last question.
If you could spawn one CRISPR system that like trumps the current limitations of the other, which would it be and why?
Yeah. Well, I'm biased, but I would say it's our NanoCast system, and largely just because pretty much everything we test about it addresses a lot of the concern. Off targets, the safety of the system when we're talking about regulatory is definitely one of the I think things that kind of gets brushed under the rug, and especially in ex vivo applications. And I think Cas9 is definitely pretty sloppy in terms of where it cuts in the genome, and that's what scares us about CRISPR, is that you're making any unintended consequences. So the NanoCast system, not only is it a third of the size, but also has the specificity advantage, has the ability to work in all these CRISPR Plus applications. But that said, I still think that repertoire approach is pretty critical towards addressing the diversity of genetic disease that's out there.
Well, thank you all, guys. This was great. Thank you, Lucas.
Yeah.
And yep, we're out of time right now, but you know where to find us for any questions, any follow-ups. I can be reached at my first name at Mayfield, and Lucas is Lucas at Mammoth.bio.
Yep.
Yep.
Yeah.
All right. Thanks, guys.
Okay.